Good afternoon, everyone. My name is Gregers Andersen, and I would like to welcome you to this Danish Diabetes Academy webinar. I'm a senior researcher at Steno Diabetes Center Copenhagen, and I will be the chair of this webinar. It's my great pleasure to introduce today's speaker, Professor Jonathan Wells. Jonathan is a professor of anthropology and pediatric nutrition at UCL Great Ormond Street, the Institute of Child Health at University College London. So I came to know Jonathan 14 years ago through his extensive research experience in pediatric body composition and energy metabolism. So we were setting up a study in Ethiopia, and we wanted to measure body composition in 600 newborns. And very few people felt that that was a very good idea, but Jonathan was one of the few that did, and he came down to Jimma to help us setting up state-of-the-art methods in this relatively challenging environment. And with his help, we succeeded and continued to collaborate on that study and other studies. And we're now planning the 15 years follow-up of those 600 children. So thank you for that, Jonathan. So apart from being an expert in pediatric body composition and metabolism, Jonathan has devoted his time to apply evolutionary life history perspectives to understand the variability in growth and metabolism. And this has resulted in the development, among other things, of the capacity load model of non-communicable diseases, which I think we will hear more about today. He has authored several books on the topic, loads of papers, and more recently, a Lancet series on the double burden of malnutrition. Jonathan has recently received a DDA visiting professorship, which we are very grateful for, and we look forward to host him at Steno Diabetes Center Copenhagen later this year. And during his visit, I think, or among other things, we will try to apply some of his life history models on some of the Danish national registers, which I'm very much looking forward to. Today, Jonathan will tell us how life history theory can be used to understand variability in human growth and metabolism and behavior. So a very warm welcome, Jonathan.
Thank you. So, hopefully my screen has shared?
Yes, you can share your screen now.
Sorry, let me. All right, that should be it. Okay, well, thank you very much for the kind introduction. So what I'm going to do today in this talk is to try and show you why I think life history theory has something extra and unique to bring to our understanding of diseases such as diabetes. So I'm going to start in the past with the Egyptian Middle Kingdom. Now, I'm no expert in the history of Egypt, but the Middle Kingdom was if you like, a golden age in the history of Egypt. It was a time of economic and social and political stability when the trade and arts and literature all flourished. So you might think that things were going very well, and that's the image we get in the left-hand side here. However, not everyone was actually very healthy. I can't advance my slide. No. Oh, here we go. So this is a skeleton that dates from this period in Egypt, and it's in fact the strongest single example of evidence from the archaeological record of someone who is likely to have had type 2 diabetes. Amongst the traits that you can see on this individual are periodontal disease, dental caries, bilateral metatarsophalangeal amputations, and diffuse idiopathic skeletal hyperostosis, a fusion of the vertebrae. Now, there's no single skeletal marker of diabetes, but all of these are correlated with diabetes. And so it looks like that this is an individual who was very likely to have had type 2 diabetes in the past. Now, I think we often think that diabetes, or type 2 diabetes in particular, is a modern disease, and that's indicated by the escalating numbers who show this condition worldwide. And we know when we study modern populations that there are a number of risk factors. And amongst the particularly important risk factors are overweight and obesity, sedentary behavior, consumption of unhealthy diets, and in particular, products such as sugar-sweetened beverages. So these are undoubtedly something that's very modern. But if we think more deeply about diabetes, actually most of these risk factors are embedded in axes of behavioral or physiological plasticity that are not overtly pathological. It's always about being at the upper or lower end of the range of a particular trait, which becomes a diabetes risk trait. So we need to increase our understanding of why just being high or low for some trait adds up to diabetes risk. And that's why I want to bring in this evolutionary perspective to try and understand our biology in more detail. Now, often when people adopt an evolutionary perspective to humans, they're interested in the traits that we all have in common. We're upright walking bipedal apes, and we have very large brains, very sophisticated communication, and having freed our hands from locomotion, we are also very dexterous. And when we try and understand why we have these traits, we look back to our past environment. So we generally assume that these traits were some kind of adaptive solutions that emerged in past environments that shaped us. Now, over the last 5 million years ago, data on the paleoclimate from oxygen isotope data from glaciers and ice sheets have shown that actually the world became steadily cooler. And during this period, the savanna niche spread in sub-Saharan Africa, and that was the niche that early hominins exploited and subsequently evolved in. And so we generally assume that these traits of having a large brain and being a bipedal animal were some kind of adaptive solutions to the challenges posed by these new savanna environments. And so that brings us to the concept of adaptation, the idea that any population of organisms has genetic variability. That variability is exposed to ecological stresses. Some individuals have better rates of survival and reproduction, and that leads to the evolution of fitter genotypes for that particular habitat. Sorry, I'm having problems with, okay, maybe I'll advance slides differently. However, we also know that in the last 150,000 years, humans have left Africa and have migrated all around the world, and they've therefore exposed themselves to many different kinds of environment. And we can use that perspective to understand the genetic basis of diabetes. For example, here are just a couple of SNPs associated with diabetes, and you can see that the prevalence in different populations is actually strongly associated with the distance of migration from Africa. So that's one way that we could investigate the evolution of diabetic risk, but it's not the approach I'm going to focus on here. In fact, if we look at the total amount of human genetic variability, it's surprisingly small. This slide uses these trees to express the total amount of genetic variability. The bigger the tree, the greater the level of genetic differentiation. And surprisingly, the sum total of all modern humans show less genetic differentiation than just chimpanzees, who occupy obviously a much smaller location and range of habitats in Africa. So somehow humans have coped with all these very different habitats and selective pressures, not primarily through local genetic adaptation. Well, why would that be so? Another stress that emerged during the last 5 million years was beyond just changes in systematic climate, also oscillations in climate. And as you can see here, from 3 million years onwards, these oscillations in climate over time got larger and larger. And these oscillations in climate translate directly into ecosystem change. And as the oscillations get bigger, what that means is that the ecosystem would likely to have been changing not only across generations, but even within the lifespan of individual humans. In other words, what we're actually experiencing today as we become more aware of climate change within our own lifetimes. Now, when we evolve in volatile environments, the process of adaptation has to become rather different. It's not possible simply to evolve a specific genetic trait that's a solution. Rather, what an organism needs to evolve is the ability to manage ecological risks. In other words, to evolve the capacity for plasticity. And then organisms can buffer a wide range of stresses through different components of flexibility, biological flexibility. And this is very much the human story. Now, we know that plasticity is extremely important for understanding chronic diseases such as diabetes. When we think of trying to improve adult health, either by promoting a healthier lifestyle or sometimes through pharmaceutical drugs, we're essentially drawing on our plasticity to improve our health. In addition to that, research over the last 3 decades has really clarified that there's also a very important component of plasticity in early life. We give particular attention to the first 1,000 days, but actually we have different axes of biological plasticity throughout the life course. And that means there's always something we can do to improve health. But once again, why do we have this plasticity? How did it evolve? And therefore, how is it wrapped up in the etiology of diabetes risk? So to study how organisms adapt, evolutionary biologists draw on something called life history theory. You can use this theory to study genetic adaptation, but importantly, you can also use it to study plastic responses, and therefore it's got a much wider applicability to understanding phenotypic variability in general. Life history theory assumes that all organisms have to solve a series of problems. How fast to live their life, in other words, what's their likely life expectancy, when they should stop growing, and when they should start reproducing, and how many offspring to produce and how much to invest in each of them. But more broadly, life history theory assumes that organisms have to solve every problem and allocate their resources in ways that maximize reproductive fitness. When it was first developed, life history theory was intended to explain the differences that we see across species. For example, why do elephants typically live for 60 years in the wild, and yet mice living in the same habitat typically live for less than 1 year of age? So we've got this continuum of fast or slow life histories with individual species distributed along that continuum. Now, why would organisms end up with different lifespans? Well, life history theory assumes that all organisms receive energy through their diet and then have to allocate it in competition between different functions. Originally, 3 functions were specified: maintenance, keeping the body in good condition via homeostasis and defending against threats; growing bigger; and reproducing. And the idea of the theory is that if you allocate more energy to any one of these functions, there must be less energy to allocate to any of the others. But increasingly, I would argue that defense should actually be seen as a separate life history function. When we allocate resources to either immune function or the stress response, it tends to be very detrimental for the other life history functions. It's not good for homeostasis, it impairs growth in children, and it tends to curtail or shut down entirely reproductive function. So together with my colleague Jay Stock, who's now based in Western University in Canada, we argued recently that defense should really be seen as its own life history function. And that's important for when we want to explore diseases such as diabetes. So we think there are 4 life history functions. And again, we're looking at the trade-offs between them. So for example, if we have to deal with a threat, either activating a stress response or combating pathogens, that reduces the energy available for other functions. So let's consider how this impacts the phenotype of different species. So here's an organism such as a mouse, and the mouse has to solve the question of how much energy to invest in its maintenance. Now, as you can see on the graph, there's a very steep inverse association between extrinsic mortality risk and the optimum level of investing in maintenance. So the mouse has a problem. A lot of other animals are going to try and eat it. And therefore, the likelihood of the mouse surviving for a long time is very low. On that basis, it wouldn't get much return from investing in its maintenance. So its optimum maintenance level is very low. Instead, to the extent that the mouse can gain energy through its diet, it should invest in reproduction. And that's the way it's likely to maximize its reproductive fitness. But supposing there was a reduction in extrinsic mortality stress, supposing all the predators vanished, now things would look very different from the mouse. If it were to invest more in its maintenance, it would be likely to live for longer. It wouldn't be predated, but it would also be expanding its lifespan. And so we can see that the optimum maintenance level would increase. And over time, what you would effectively see is that mouse realize its aspirations and evolve into a larger, longer-lived animal, such as a meerkat or a marmot. So we can use exactly the same approach to study trade-offs during the life course. As I mentioned, if we develop an infection, we have to allocate more resources to immune function, and that will help get rid of the pathogen, but it will also reduce the energy availability for these other life history functions. So that gives us this second concept of adaptation. We have phenotypic plasticity, many different axes of biological plasticity, including behavior. We expose that plasticity to ecological stresses, and we make fitness enhancing responses in the short term, and overall, those should lead to an adapted phenotype. Now, if we want to study these trade-offs in humans, we can't just use observational studies. We can't just look to see, for example, if children who have infections are shorter. This is what I call the mansion, limousine, and yacht problem. Some individuals, in economic terms, are just very wealthy. They can afford to live in a mansion, but they can also have a really nice car, and they can own a yacht. So we won't see what the stresses are of owning a mansion if we just look for correlations. What we actually need to do is an experiment. We need to force someone to buy a second mansion, and to see if to afford that, they then have to sell one of their limousines or their yachts. So we need experiments to really reveal life history trade-offs effectively. So how would we do this? Well, obviously, many people do experiments on animals because animals are very easy to control and you can do very precise protocols. Many people do research on fruit flies or rodents. Sometimes for humans, we're interested in species that share some of our traits. Seabirds have quite long lifespans, and primates, of course, have quite large brains. And here's an example of a study that really reveals very nicely trade-offs in an animal species in relation to some kind of environmental stress. This was the amount of protein in the diet, and it was a study of Drosophila fruit flies. What you can see very simply here is that a low amount of protein in the diet will maximize the lifespan of the fly, but a high amount of protein eaten will maximize the lifetime egg production and the egg production rate. So in other words, there's a trade-off for these fruit flies between reproducing and living a long life. And that trade-off is determined by the amount of protein in the diet. You can either live long or you can reproduce a lot, but you can't do both. Well, what would we do for humans? So it's quite difficult to give humans severe experimental protocols that would induce some of the stresses that we would most like to study in order to really unpick the adaptive components of human metabolism. But together with my colleagues in Cambridge, Jay Stock and Danny Longman, we decided to use athletes as a new kind of animal model to research life history theory. And the rationale was that athletes voluntarily, regularly put themselves through really quite extreme stresses. And that allows us to do quite controlled studies and to look at big changes in energy supply or expenditure and their associations with life history outcomes. You can think of every body, humans in particular, as playing an energy allocation game. We get a calorie in the diet and we have to decide what to do with it. We could either expend it on a function, maintaining the body, synthesizing new tissue, physical activity and muscular effort or reproduction, or we could store the calorie in one of several tissues, either organs or muscle or a fuel depot, which could be glycogen or adipose tissue. And these decisions as to how to use the calorie are enacted at the first level by hormones, such as insulin, leptin, or adiponectin, although we could get much more sophisticated and look at metabolomics and other forms of biochemistry. So what we wanted to do was to use some experiments that would actually force humans to produce different results in the energy allocation game. So the first study, led by Danny Longman, was on 62 young men who were all college rowers. And these men were randomized, first of all, to do one of two tasks separately: either row as hard as they could on a rowing ergometer, or to participate in a simple memory test. So they did those on two separate occasions. And then on a third occasion, they came in and they had to do the two activities at the same time. And the idea is that we were either challenging the muscles or the brain, or the muscle and the brain together. And the question was, if we challenge them together, which one would be more effective? And these are the results. You can see that both cognitive recall and muscular output declined when the athletes were obliged to perform the functions simultaneously. but the power output declined more than the brain. So this indicates that when you force the brain and the muscles to compete for energy, the brain will to some extent win. And that has direct associations with the thrifty phenotype hypothesis and the concept of brain sparing in early life. If you force the fetus to compete amongst itself for energy, it will protect the brain at the expense of the muscles. Here we're just seeing that on a moment-by-moment basis. A second experiment that we did looked at a much longer exposure to sustained energetic stress. So this study was conducted on 66 heterosexual athletes, and they were taking part in an ultra-endurance marathon, the North Downs Way in the south of England. And this is a 165-kilometer race. The course record is 15 hours and 3/4. So obviously, most athletes are taking 16 hours or longer to complete this race. And we collected some very simple measurements at baseline and at the end of the race. We collected two markers of reproduction. One were salivary measurements of the hormone testosterone, and the other was an index of arousal. We simply showed the athletes pictures of erotic images, and we asked them on a visual scale how aroused they felt. Then we collected two markers of the body's investment in immune defense, hemolytic complement assay and bacteria killing assay. And as you can see here, over the course of the race, effectively the body withdrew its investment in reproduction because testosterone and arousal went down. And instead, the body started shifting resources to protecting itself against potential pathogens. So both the hemolytic complement assay and bacteria killing assay increased. So under sustained energetic stress, the body shifts its allocation of resources from reproduction to survival. Now, the third study that we did wanted to have a look at social cues while controlling for energy stress. So this time we did a study of 19 pairs of adult males, and these men were lined up together on rowing ergometers, and they had to row as hard as they could for 10 minutes nonstop. However, we covered up the display on the machine so that they couldn't actually see how they were doing. And then we randomly told one of them that they had won and the other one that they had lost. So the idea here was that the energy stress was broadly similar between the two men in each pair. However, the big exposure was that we randomly explained that one was a winner and one was a loser. So we had detached the social experience of losing from any energetic stress that might have led to winning or losing. We have no idea who actually rowed faster in each pair of men. When we looked at the outcomes, we found that the winners experienced a spike in testosterone, whereas the losers experienced a decline. And when they were given a behavioral questionnaire, the winners felt more confident about approaching a woman for a date. So there's also a behavioral marker of the willingness to invest in reproduction, although obviously it's a hypothetical situation. So here, what we've shown is that life history trade-offs can occur in response to social cues that are detached from any underlying energetic investment or activity. Okay, so those were some studies of athletes, but we also are still very interested in the stress response itself. And so for that, we needed a slightly different way to do experiments. And the idea here is that activating the stress response would reduce the ability of the body to allocate energy to other functions. So this time, we wanted once again to select a stress that people participate in voluntarily. And so we used video games. And as you can see from these photos, people really get very intensely into video games. It's not what you'd call an active pastime, but certainly these individuals are very active in their imagination, and that's quite important. Now, most video games involve some or other kind of task or competition. And one of the most popular video games is the FIFA World Cup game, so a sporting competition. However, it doesn't take long if you look at video games to realize that many of them involve extreme levels of violence, and these games are very popular. So we thought we would look in more detail at video games by actually trying to see both what video games per se do to the stress response and also if the violent games do something different to the non-violent games. We initially did just an epidemiological small study on video gamers per se. And it wasn't difficult to see that people who regularly play video games have a number of traits that we would regard as risk factors for a condition such as diabetes. The more they play video games, the more these young men have higher BMI, higher waist girth and fat mass and heart rate. They tend to have a diet with greater levels of added sugar and low levels of fiber. They show lower levels of cognitive restraint, and they show low levels of physical activity. So just looking at the profile of video gamers per se, they don't stand out as a particularly healthy group in the study. So we did a couple of randomized trials. The first one was of young men of normal BMI, so below 25 kilograms per meter squared. We gave them baseline measurements of the stress response. Then we randomized them to either watch a comedy show on TV, or play the FIFA sports game, or to play a violent video game. Then we monitored the stress response for 1 hour during the intervention and collected end-line measurements. And these were the results of the first study. So compared to the TV controls and the non-violent video game group, the group who played the violent video game showed higher diastolic blood pressure through the intervention, and they also felt more hungry. Now, we interpreted that there were higher rates of cerebral glucose uptake in response to the stress of violence, and that was generating, in response, greater feelings of hunger. But we couldn't prove that because we didn't actually have any measurements of energy intake. So we thought we'd repeat the study on a slightly larger sample, and this time we would only recruit overweight men, having seen that many people who play video games are already overweight. And we would broadly repeat the study design, but this time we would include at the end of the intervention a test meal. So we would provide some foods and see what people ate and how many calories. Now this time there was no difference in the stress response markers between the two types of video game. If you grouped all of the video game players together, then they showed higher blood pressure compared to the TV watching controls, and using a visual analog scale, they reported that they felt more stressed while playing the game, although that resolved at the end of the intervention. But when we looked at the test meal, while the group of video game players combined showed greater intake of sweet foods and saturated fat compared to the TV watching controls, when we looked at the two video game groups separately, it was only the group who played the violent game that actually consumed more sweet food and more saturated fat and more calories compared to the TV watching controls. So there does appear to be an interaction between the stress specifically of violence and the effect of playing video games on appetite. So we think this is quite important and can help understand the epidemiological data that consistently relates exposure to stress with higher levels of BMI. It's a slightly different model of obesity to calorie counting. We're not just saying how many calories are going in, we're asking why they're going in, and we've called it a metabolic perturbation model. We're trying to understand not only why we eat, but why some of us persistently overeat. Of course, a limitation of these studies is that our measurements of appetite were short-term, and we didn't investigate whether those who ate more after the video game might have ate less later in the day. So I hope what you've seen so far is that we can start to improve our understanding of some of the risk traits that are associated with the global emerging diabetes epidemic. Now, we tend to think this is a modern thing. As I mentioned earlier, the numbers are going up now, and we connect it with this broad term, the nutrition transition. People are less active, they've got more modern lifestyles, they're eating more food and different foods, and that everything about that seems that this is a modern disease epidemic. But actually, this is not the first nutrition transition that humans have experienced in their history. So we have another classic nutrition transition, which is the shift by the vast majority of human populations at some time or other from a hunter-gatherer lifestyle to the adoption of different forms of agriculture, when many aspects of living changed. Now, we have some rather surprising things when we look back at the archaeological record of agriculture, which have long perplexed archaeologists. On the one hand, there's no doubt that the global human population systematically increased when agriculture was adopted. So in that sense, from an evolutionary point of view, we would say humans increased their reproductive fitness. but the quality of human biology appears to have gone down almost systematically. For example, many studies have shown a decrease in height following the adoption of agriculture. Here are some of our own data looking at the Indian archaeological record. And although those data are expressed in Z-scores for bone lengths, they translate into a reduction over the last 6,000 years or so of around 15 centimeters in adult male height. So that's a really big decline. Then there are increased signs of markers of bone disease, such as tuberculosis in the spine, shown here in evidence from Peru. And very occasionally, when we're able to analyze data from Egyptian mummies or mummies from other populations, there are some signs of arterial disease as well. So more humans, but less healthy humans. How can we explain that? So once again, we think that life history theory may be able to offer an example. And this was another paper that I published with my colleague Jay Stock last year. Looking in more detail about agriculture, it often involved changes in subsistence practices, for example, grain harvesting, but also food processing, making the release of energy more efficient. And in many populations, the emergence of dairying, allowing us to eat dairy products, or in some cases, also to drink milk directly. But also, agriculture involved populations becoming more sedentary, and that had many outcomes in its own right. Village living increased the exposure to pathogens and also reduced the length of the birth interval. So overall, humans who began farming tended to have generally increased energy intakes, but also paradoxically an increased risk of famine. And humans were also very good at exposing themselves to their own stresses, the pathogens that benefited from more dense human populations, and the fact that human populations often attacked each other and tried to steal each other's harvests. So overall, what we think we can see in the archaeological record is markers of increased energy allocation to reproduction, shown by the rising fertility, more energy allocation to defense, shown by people who are alive, but showing symptoms of disease in their bones, and therefore less allocation to maintenance and growth. And again, we can see the reduced growth in the reduced adult stature. So this gives us the idea that we could once again use life history theory productively to improve our understanding of why health isn't necessarily universally increasing in association with nutrition transition in modern populations. Now, finally, I've talked so far about life history trade-offs within a generation, but we can also look across generations. All of the life history trade-offs made by the fetus and infant in its own early life ultimately relate to the supply of energy provided by the mother. So the mother's budget for reproduction determines the energy supply for the offspring, and the offspring then has to make its own adaptive trade-offs. So one of the studies that we have been working on is to see if we can change these trade-offs in early life. And we chose once again to look at the stress response. Now, it's very clear when working with mothers that even just being a mother has a range of elements of stress. Mothers have the new responsibility of looking after the infant. And interestingly, lactation itself is not instinctive, and mothers often worry about whether they're breastfeeding adequately. So we hypothesized that even amongst healthy first-time mothers, we might be able to benefit them by reducing their stress. So we hypothesized that if we could reduce the energy allocation to stress in mothers, that would therefore increase the availability of energy for the other life history functions. And one of the benefits of that might be a change in the supply of milk to the breastfed baby. So one of our PhD students, Husna Mohd Shukri, conducted a randomized trial in Malaysia. She worked with 64 healthy first-time mothers and their babies. 32 were allocated to a placebo group of just standard support for breastfeeding, and the others were allocated to the intervention group where they had relaxation therapy. And this was very simple. The mothers were just given an audio cassette, which they were encouraged to listen to while breastfeeding, and the audio cassette encouraged them to feel good about themselves and to relax. And then the student went 3 times to the homes of these families and collected data. Now, I don't want to go into the detail because time is short, but essentially this relaxation therapy completely rebalanced energetics across both the mother and the baby. The mother reported feeling less anxious, and she produced significantly more milk, measured by stable isotopes. And there was also a change in the biochemical macronutrient composition of the breast milk in relation to the carbohydrate content. The baby slept significantly longer, but most importantly, the baby gained significantly more weight. Now, this is fascinating because we gave no more calories into this system. All we tried to do was to change how the mother was using her calories within the body. She may have had increased appetite and eaten more, but that wasn't the intervention that we directly introduced. So I think this is very exciting in showing how we can potentially change life history trade-offs in 2 generations, again, through a behavioral intervention. So overall, what I've tried to show you in this brief presentation is that in our life history studies, we've been manipulating on the basis of theory a number of traits which actually have clear implications for the etiology of diabetes risk: muscular effort, the hormone testosterone, the stress response, immune defense, appetite, and even birth weight. All of these are known correlates of diabetes risk. But what I hope I've shown you is that we've got more understanding of why these traits vary in response to ecological stresses. And if we can build on that knowledge, we might be able to develop more successful interventions. So I'm really extremely excited about my opportunity to spend time in Copenhagen, funded by the Danish Diabetes Academy. As Greg has said, we've known each other for nearly 15 years. I've been very privileged to work with him on the IAPC cohort in Ethiopia. It's a really unique study. I think it's producing unique data on the etiology of diabetes risk in a low-income setting. And that's very relevant to European populations as well because of international migration. And it's also been a particularly enjoyable study to collaborate on. While I spend time in Copenhagen, I'm hoping to contribute to 4 different components of work. In each case, partly just contributing to the epidemiology or cohort studies, but also trying to bring in the life course evolutionary perspective to help understand why things are going as they are. And the 4 studies are studying the life course etiology of type 2 diabetes risk in the inter-99 cohort of older adults, looking at the association of childhood stress and subsequent trade-offs in the Dan life cohort of younger adults, improving understanding of the etiology of diabetes risk in low-income settings, in particular the IABC cohort in Ethiopia, and also improving our understanding of how climate shapes diabetes risk in Greenland through work on 2 birth cohorts there. So I'd like to acknowledge people who've been particularly important in my developing this application and the studies that I've described here. And I'm very happy to answer questions if you have any. Thank you very much.
So thank you very much, Jonathan, for this very inspiring and mind-blowing presentation. I think it pushes or rocks the way we are thinking about diabetes and diabetes development. We have only a few minutes left, but if there's anyone with a question from the audience, you're most welcome. So I just give, Christian Mølgaard.
Yeah, thank you very much, Jonathan. Very interesting, as always, and also very complicated. But now we have a big pandemic, and if you look, go back in history, there have been several pandemics, and also in the Middle Age for some, and also longer below earlier. How do you see this kind of events, or what you can call it, in relation to your ideas here?
So some of the markers that we're interested in, you could clearly find evidence for. So the discipline of anthropometric history looks to understand long-term changes in human height. And I think in the 20th century, the influenza epidemic was shown to leave a signature in later height. And so did the Ukraine famine, and the Ukraine famine has been directly associated with diabetes risk. So even if the evidence is poorer in the medieval period, I think we can expect many of these responses to have happened. Whether people actually went on to develop overt diabetes is a much harder question. Certainly they would have started to accumulate diabetes risk factors, but there is a difference between having the risk and actually having the disease. And that's something that we need to understand in more detail. I mean, today it's closely associated also with developing overweight as an adult, but many of the components of risk would have been there.
Thank you.
So let me see if there are other questions in the audience. Yes, Jose.
Yes, hi. Thank you very much for the presentation. That was very interesting. I was not familiar with your work, so that was a really nice way of getting an introduction to your work. I'm Jose Areta from Liverpool John Moores University, and we do mainly work with athletic population, but not only. And we work around the concept of energy availability a lot, which, I mean, you mentioned energy availability a few times. And we're trying to understand how decreasing energy availability sort of affects different sort of systems and tissues and so on. So we have a few lines of research in this area, and you might be familiar that energy availability is considered like the etiology of the probably more popular female athlete triads, where decreasing energy availability, you know, affects reproductive function and bone metabolism and so on. How do you see this fitting with this particular model?
So I think the opportunity to examine sex differences is particularly exciting. Exactly as you say, we shouldn't necessarily expect the two sexes to respond equally either in their metabolism or their behavior. And actually, probably the most helpful response I can draw on here is to go back to other work I've been involved with through Copenhagen, which is studies of how men and women respond to nutritional supplementation during treatment for TB. And each sex loses the tissue that is important for reproduction. Men lose lean mass, women lose fat mass. And when they regain weight after recovery, they gain back what they lost. Reproduction is a luxury. So the first thing you give up under energy stress is what is a luxury, which is essentially reproduction. So men lose some of that upper body muscle mass and women lose some fat mass. So I think what you're referring to there is actually another great opportunity of life history theory to shed light on why these sex differences are there and understand sometimes why the pathway to diabetes may be different in men and women.
Yeah, thank you so much. And one last question. Where do you see the energetic stress or like locomotion fitting in the model that you presented?
There's a continuous debate about how active humans have been through their evolutionary history. And I think sometimes we picture that we were all extremely active, and actually hunter-gatherers are not necessarily so different from us. They're not always vigorously active, but they're almost never as sedentary as modern humans. So we could study persistent energy expenditure tasks and improve understanding of that. I think you would need to study people doing different levels of exercise and actually look at the consequences.
So, thank you.
Okay, so once again, thank you very, very much, Jonathan, for this very inspiring presentation, and we very much look forward to receive you, welcome you here in Copenhagen later this year.